And today I would like to give a brief story about inline process analytics for advanced therapies. Before jumping into the actual content, I want to also position ourselves a bit and what we are doing. So everyone knows Johnson & Johnson, mostly because of a few blockbuster drugs that are currently on the market, mostly coming from the antibody field. However, in the last decade, we have ventured also in the, let's say, the next generation therapeutics field with quite some success at this point in cell and gene therapies. And we are here, process, biophysics, and process analytics. And as you can see, we are kind of squeezed in between our folks from discovery, which is the entry point, obviously, and then the manufacturing science and technology, which is the exit point. And we work together with our drug substance, or API, and then drug product, DPD colleagues, and we help them developing solutions to better control their processes and also to come up with smart analytical tools to monitor these things, preferably in real time or as close to real time as possible, and mainly focused on product characterization and process analytics. But of course, before talking about our ability to stay inline and online, one first has to learn to do some sort of a freestyler offline dancing. And I want to go through a bit of history. Like in 2010, I joined the one and only company. So not only Waters have these long-standing employees, but also Johnson & Johnson. So people are being around for quite a lot of time, from the Paleolithic, as my colleagues would like to say. And in 2010, the first mission in discovery settings was to set up a laboratory to kind of have a choro, like we kind of have a grip on our molecules to know exactly what we are dealing with. And first few days in office or in the lab, I was presented with this situation where people were furious that the columns, the size exclusion columns, are just crap. This is an antibody injected on a size exclusion chromatography. They were saying, look, we just cannot make this reproducible. So how are we going to solve this? And at that point, in addition to this totally outside the box distribution of sizes that was seen on the size exclusion chromatography, let's say none of the conditions would kind of lead to would drive the main peak to the desired retention time, retention volume. So based on this, you would conclude that pretty much all our antibodies are like bits and pieces of an antibody, right? So this is kind of the dilemma I had there. Shall I stay or shall I go? But then also based on what we know about size exclusion chromatography, we know that elution in size exclusion chromatography is determined by size, not mass. We know that secondary interactions could happen in size exclusion chromatography. We know that aggregates can dissociate on the column, they can be induced on the column. So in a way, it's kind of like, it's a very nice puzzle to solve if one would be ever able to solve it without some extra help. So let's start from the beginning. This is a tau protein. It's one of the culprits associated with Alzheimer's disease. In its cellular form, it's supposed to stabilize microtubules, and it comes in 6 isoforms. This is the longest isoform with 2 N-terminal inserts and 4 microtubule binding units. And if we run the highest resolution in analytics, SDS page, the protein is fine, right? You get the right band, so you're celebrating. And then why not, let's just to make sure that we are right, do also size exclusion chromatography. And again, we see a beautiful, big, homogeneous by eye. The only problem is that based on this positioning, you would tell that this protein has a molecular weight of 170 kilodaltons. Now that would correspond more or less to a trimer. Now, like I said before, this is one of the culprits in Alzheimer's, and the mechanism of pathogenesis in Alzheimer's is not really elucidated. So at this point, there are only 2 options. Either we are on the path to get the Nobel Prize, to kind of finding the critical nucleus that's responsible for aggregation, or we don't know what we are doing. Now, But then we remember size and mass are not the same thing. So tau protein, highly unstructured. IgG1 protein, not quite unstructured, quite packed. They both have a hydrodynamic radius of 5.5 nanometers. However, tau protein is 50 kilodaltons, whereas IgG is 150 kilodaltons. And we just remember that size and mass are not necessarily the same thing. So since they have the same hydrodynamic radius, there is no surprise that tau protein elutes the same position as an IgG would elute, which corresponds to molecular weight of more or less 150 kilodaltons, right? So unstructured proteins have a larger hydrodynamic volume than the corresponding to globular proteins, so that explains the earlier elution. So what's the solution? Well, this morning was quite an extensive presentation on this. In a nutshell, particles, when they are irradiated by a laser, scatter light. This light scattering is proportional with molecular weight, concentration, and also with the differential refractive index, which is a property of specific material. Scattering happens in angular manner above a certain size, but with extrapolation to angle 0, one can actually calculate the molecular weight as long as the intensity of scattered light is known, the concentration is known, and the dN over dc is known, right? Going back, what we see here on the right side is exactly what I've showed you in the few slides back. The only difference is that now we're also going to factor in what you can get from a light scattering detector, which kind of, by measuring this, the molecules within this peak, leads us to a result which is actually very close to a monomeric protein. Good news, we know now that tau is a monomeric. Bad news, we are not going to get any Nobel Prize. Now, that thing has been ticked off, right? Now we have a direct way of measuring proteins and determining their molecular weight and aggregation status. Still, the relationship between column and sample, it's like most relationships, not quite straightforward. So what is our approach here? This is what we do normally when we have samples. We like to kind of zoom in, see exactly what are the components there. We have, like, let's say, some sort of a higher resolution, a separation, which we do with size exclusion chromatography coupled with multi-angle light scattering. But in the same time, we want to know that the sum of all these components that we see upon separation adds up to the total, right? For which we perform always, before injection, a dynamic light scattering experiment in batch mode. So in a metaphor, first we want to see that roughly we have a blurred picture, but we kind of know that we are dealing with apples, and all apples are mostly red and have kind of sizes within certain ranges. But then if you want to find out details, we want to also kind of like zoom in, have a more clear picture, see what the sizes are. There are some differences. We might identify once in a while also some rotten apples, but that way we can actually tell that what we see in a higher resolution picture is not unexpected. In sum, for the introduction, the multi-angle light scattering replaces speculations with facts by directly measuring molecular weights. All size exclusion measurements coupled with multi-angle light scattering should be interpreted in a broader context, including batch size measurements and also recoveries from the sample. So we want to know that what we injected, it's coming back. Otherwise, getting a peak when you only recover 90% of your sample could mean anything. And in conclusion, if all of the above are happening, SecMoS will separate the big problems from the small talk. Now, shifting gears towards process analytical technologies and cell and gene therapy. Now, cell and gene therapy is a market quite expensive, you already know from this morning, but it's estimated to be around $5 billion Anno Domini 2024, expected to reach around 50 billion by early 2030s. Large numbers of patients are waiting for this treatment, so at this point, really, the industry is the bottleneck. We have a product that, theoretically, if you would be able to produce a certain rhythm and all the logistics in place, we could probably make way more money than we are currently making. but we are struggling there. Treatments are extremely expensive, and these molecules are very complex. So if you think about a protein, it's a protein. You know exactly the sequence. Of course, you have to do the quality control, but you know exactly what you're dealing with. In these cases, we have an assembly of these molecules are assembled from nucleic acids, a whole bunch of proteins, glycosylation, complexity, etc., etc. Now, I don't have to tell anyone that a 1% increase in efficiency in a $5 billion or $50 billion market is actually quite a lot of money. So PAT, Process Analytical Technologies Initiative of the FDA, has been launched a while ago. Nice words. We are mostly interested in the content, less in the words, but of course the words are also nice. But the bottom line is the real-time monitoring offers timely determination of critical quality attributes and rapid feedback on product characteristics. It helps in process optimization. Continuous data enables better process control. So we know exactly what is happening in real time rather than, like in the morning it was mentioned that it could take up to 4 to 6 weeks till the process gets the feedback from the analytical department about what's up. Not to mention the efficiency per se, like we don't need to collect samples, we don't have handle, we don't have to involve millions of people, not millions, but like quite a lot of people in testing, etc., etc. And the enhanced product quality enables quick detection of potential deviations and issues. Nice words. How is it done? So what do we do here? And this would also solve the relationship between column and sample because you don't have to deal with the offline columns anymore. So this is not a random process that we have an instrument and let's use it in line. Actually, there are quite some complications. And in finding the right technologies, we go through a certain process. When we are looking first of all, in what type of molecule are we dealing with? Advanced therapies could mean a lot of things, right? There are some standards at this point that people are thinking about when we talk about advanced therapies, but just think about an AAV is already, in terms of hydrodynamic radius, let's say 3 times larger than an antibody, but that means it's 30 times larger actually in terms of volume. If you think about the lentivirus, that is kind of 1,000 times larger than a protein. It's extremely complex. The components in the protein, in the molecule, again, the distribution of proteins versus nucleic acids. If you talk about the RNAs, in that case, you don't have a theoretical model how it should look like, but you encapsulate the RNA lipid nanoparticles, and then again, good luck with that as well. It's very important to be aware of the matrices. There is one thing to deal with process analytics for upstream processing, and a totally different thing to deal with process analytics in fill finish. What are the critical quality attributes to be monitored? What are the critical process parameters, the critical process attributes? What is the sensitivity? This is very practical, but very important. What is the sensitivity, the frequency needed for the measurement? Do you only want to know, like, I don't know, every day to measure a titer, to know that you are moving in the right direction, or you would like to see how the host cell proteins accumulate on a continuous basis? How quickly do you need the data? So do you need to process whatever you're looking at? Then we just move the bottleneck further, or you can do it right away. Regulatory requirements, and of course, what are the available technologies? And there we are in desperate need for more. So what we want to do is to bring the process as close as possible, if possible in line, which means that we measure on the fly, so we know in real time what is happening. If that is not possible for technical reasons or limitation of technology, we like to do online, we do bypasses and auxiliary pumps and so on and so forth. And the worst case scenario at line. But we would like to forget as soon as possible about offline, where the laboratory is somewhere else and we ship samples and we wait. This is our toolbox. I'm not going to go into detail because it will take too much time, but pretty much we are dealing with a whole bunch of different active components. We have different stages in process development, starting from extremely dirty, quote unquote, towards drug product development when the sample becomes cleaner and cleaner. And these are a bunch of the technologies we are currently using in our department. And like I said, we're exploring some others, and we're in desperate need for more, preferably to be able to monitor as much as possible, if not everything that's possible, inline. And now a story about real-time multi-angle light scattering for downstream processing of AD26, adenovector 26 viral vectors in this case. So what is the adeno26 viral vector? The adeno26 viral vector became probably the most became famous, especially during COVID vaccine development, because it has been our platform for vaccine development. And the adenovectors are complex structures with a size, a diameter around 90-95 nanometers, with a protein nucleic acid ratio of 10:1. Right? Here are some of the major proteins, core proteins, cement proteins, so to speak, and along with the copy numbers in the hundreds. And these are some cryo-EM pictures about how the structure looks like. So needless to say that if you really want to make sure this molecule is perfect, we need to dig a bit deeper than for a protein. So we're dealing already with dozens of proteins and nucleic acids. Now, this reminds me of this commercial from a while ago. I don't think the statements are fair, but that was the advertisement. It was this question about what food do they love? They love vegetables. I think it's not correct, but anyhow. And what food they hate soup. So how do we look at the vegetables in this soup? So the main thing in selecting technologies to monitor these adenovectors was that we are looking at something relatively large, very large. So the radius is around 45 nanometers. That's like 9 times larger than a antibody in terms of radius. The power of 3, dealing about 1,000 times larger particles than an antibody. An antibody is not a small protein. So if you think about something like slope technologies or some other technologies we are using based on UV or other parameters, well, you can imagine that they see everything. This matrix is just incredibly thick in UV. We are not going to be able to see what you want to see. On the other hand, using light scattering, and due to the proportionalities just mentioned before, the light scattering sees pretty much these big particles, and in this case, lucky us, it doesn't see what's around it. So if you want to monitor titer, if you want to monitor size, actually it's an excellent technology, we thought. So we don't want to see the adenovector, we want to see the adenovector, we don't want to see highly absorbing matrices. And multi-angle light scattering is responsive to the 6th power of the radius, so buffer interference is virtually filtered out. We hate the soup, we don't see it. Now, what we used here is a real-time multi-angle light scattering detector, which is developed and optimized for process analytics. It's a Ultra-Dawn, and one of the most important things here is that this detector can function as advertised up to 150 milliliters per minute. We didn't go that high, but that is a big difference between analytical detector and this one. The data density is high, it can have up to 5 measurements per second, and the response time is also fast, so we can actually accumulate data in a very fast manner. The range for molecular weights, again, we didn't reach the side, but the extremes, but supposed to be between 10 to the 9 grams per mole. Again, there is quite some range that can be used for size measurements. We can turn in particle concentrations, and with red I have this fully empty ratios, capsid and genome molecular weight, that is correct. However, there is an asterisk there for adeno that's a bit complicated because the distribution of the protein and nucleic acids is kind of one-sided. So there's not going to be a big difference in the properties of the molecule, whether it's full or empty. But this is it works perfectly fine, for example, for AAVs. First experiment, we coupled the Ultra-Dawn to purification systems, to this just an ACTA, and via the observer software. The observer software is able to actually trigger signals and calculate values, so it's kind of like ensure some sort of a feedback system that helps, especially in production, to be able to kind of start, stop, but also select what you want to see, what you don't want to see, what you want to collect, what you don't want to collect, based on the properties of the material. So let's start with the beginning. It's an anion exchange chromatography, right? And of course, in addition to the light scattering detection, we still have the traditional UV detection. In the conditioning phase, between 6 and 11 minutes, you see that actually we start seeing signal both by UV in sort of purple and light scattering. So the conditioning means that we are flushing the system without having the sample entering a column, so bypassing the column. And after 11 minutes, we know that the flush has happened, the signal is stabilized, so we can start loading. And the loading happens between 11 and 15 minutes. You see, you have actually a huge UV signal, but we have no light scattering signal, which means that actually the big particles are not coming out of the column. Actually, they are sticking to the column as they are supposed to be sticking on the column. Just think about, without a light scattering detector, at this point you don't know. It's just a lot of UV. You don't know whether UV is coming from the particles or from the buffer. And then you see right away also when the column is full, saturated, because then you see a further jump in UV, but also you start seeing actually significant light scattering. That's also good to know when it's time to stop, because this material is precious. Then we have a wash. Again, we ensure that everything is washed off between 17 and 20 minutes. So again, you see a large UV, which means that small particles and maybe fragments, could be also other materials coming out. But again, there is virtually very little light scattering, which means that actually during the wash process, we do not impact our material. Our material stays on the column. Of course, the material is on the column, then you also want to get it back because that's fair. So this is just a representation of the elution process. So there is no gimmick in the separation per se, it's more like the detection. So here you see again the elution profile both by UV and you can also measure the titer using the light scattering data. But in addition, we can also actually monitor the size of the particles that are eluting from the column. So we know that everything that comes off the column is the right size, and at least based on the physical properties of the material is the right thing. What we think also is extremely valuable is that we could actually trigger collection. So the system can get from via the software these instructions to collect certain types of particles based on preset, let's say, conditions. In our case, we set that the size cannot be smaller than 40 nanometers, cannot be larger than 50 nanometers. But we also set some criteria for a minimum light scattering that we need, so we don't dilute unnecessarily our pool of collected material. So this is the part that has been collected here between, I don't know exactly what the times were, like 24. It's actually quite narrow band. You see that we have signals also in a couple of other places, but in these cases the collection trigger was not activated because one of these two criteria was not met. Either in this case the measured size was totally crazy territories, or the intensity of the signal was quite low. This has been already mentioned. Now, if you look at the results, and for full transparency, I listed here all the repeats. So we did we used two different adenovectors and also used different flow rates. So we are still far from the claimed, let's say, upper limit for flow rate. So it seems that it can go way higher. But under our conditions, two things. So we measure the radius by real-time multi-angle light scattering, and then we compare it with offline methods, with the established offline methods, like in this case, the dynamic light scattering. And if you see that pretty much every time we see 2-3 nanometers, like a radius which is a couple of nanometers less than what we measure in dynamic light scattering, which might induce some panic initially. But then we remember that actually hydrodynamic radius and the radius of gyrations are two different things. So hydrodynamic radius is in principle a theoretical, it's the radius of a theoretical sphere that would have the same diffusion coefficient as the considered particle. When you compare it with the radius of gyration in general, it's normal that the hydrodynamic radius is always larger. And if you factor in all the theory behind light scattering, actually these results make perfect sense. Now, if you look at titer, again, the results are not necessarily right on top of each other, but we are comparing here again like a titer obtained directly, measured based on the light scattering principles, with a titer obtained from capillary zone electrophoresis. When we are having a theoretical input, we're integrating a peak based on an extinction coefficient that it's assumed. And again, some variations can be expected, but we are talking about like a 10% difference between measurements. And also we have to keep in mind at which stage we are at. So we are in downstream processing, where actually we could probably tolerate even 20-30% error, because this is not a final step. This is just a step based on which a decision is going to be made as how to proceed. Like, how much are we going to load on the next column? If it's another column, how much do we have to dilute going towards the final product? So far, we are quite happy with the results that we are getting. Now, going further in the process, this is another example that where we have UFDF stage, tangential flow filtration. In this case, the actual flow is around 600 mL per minute. So obviously we cannot insert this detector in line. So we used an auxiliary pump, a bypass, so to speak, where we actually extract samples both from the retentate and the permeate, and we send it to the detector, and this can happen every few minutes. But in this case, we are just changing buffers. So actually what we know, of course, is nice to see the size in real time, but what's even most important is to see that actually the size is constant and the titer is constant, much more than the absolute value.
Adrian, 4 minutes left.
I think I'm fine. Then if you look at radius and titer consistency, I think you can agree that pretty much the radius is incredibly constant, like during all this process. And when you look at the titer, we have like some little fluctuations here, but mostly they happen when we are connecting and disconnecting the pumps. So we think it's an artifact of like how the systems are flowing, and this happening there. But even there, we are talking about like, I don't know, like a half a, if you look at this scale, everything oscillates between 7.5, 8.5, 10 to the 11 virus particles per milliliter. And well, one more slide here, or two more slides actually. So here we are, let me just put everything up, sorry. This is the final thing, the final dilution, where we are moving from 2 liters to 14 liters. I don't know what happens to the 14 there. It's sequential, and again we are applying the same theory, and then we measure pretty much the titer at each individual step. And when we represent this titer relative to the expecting titer, we see quite some good linearity. The highest 2 points, pretty much the first 2 points in the measurement are a bit off, and we believe that what happens there is that there's quite some significant change in the refractive index as we move from the initial formulation buffer to the final one, and this happens very quickly And since there is also the intensity of scatter light is proportionally increasing with when the contrast between the particle and the medium is the refractive index is bigger. So there is some deviation there. However, again, what we are interested in actually is the final dilution, not the initial one that we already knew. But even that we can fix by normalizing, and because pretty much the intensity of scatter light is proportional with size and number of particles. And you can always find the proper calibration constant. And if you use that calibration constant, as you can see, actually we have linearity for the entire range of concentrations. And this is my summary slide. Real-time process analytics in development. It accelerates process development and scale up. We understand how process changes impact product quality in real time, reduce offline measurements. And in production, we can monitor and control the process by critical quality attributes. Indicate the process endpoint, start and stop pooling. We can detect problems in real time. And of course, we acquire knowledge. And in-depth knowledge of active component, process parameters, and technology is crucial. Sorry for running the last couple of slides. Your fault, sorry. but yeah, this is the acknowledgement. And I'm really happy that Wyatt and Waters are now in the same pocket. And I hope that size exclusion without light scattering is going to be forbidden. I don't know if you have time for questions, but
Yeah.
okay.